everyone. Good morning. It's my great pleasure to welcome Sergei Maslov to give a seminar talk today. And Sergei is a professor of bioengineering and physics at the University of Illinois Urbana-Champaign. He's also a affiliated NITMB member. His interest involves evolution, microbial ecological system, and recently applying deep learning to those systems. And I'm personally super happy that Sergey is here because I known him for a long time and he was on my thesis. And so with that, I want to give the time to Sergey and please take it away.
All right, thank you. Thank you very much for the invitation. It's a great pleasure again. Yeah, so it's not my first visit, but it's my first time in this conference room. The last time I was here was a conference which I co-organized in May, and this room was just being finished up. So we didn't, so it's gorgeous. I mean, it's hard to, you guys are not distracted by the view, but I am. So what you see here is where I am actually spend all of my working hours. This is Institute for Genomic Biology, and one of the hats I wear at UIUC is that I'm co-director of the Center for AI and Modeling. And I will kind of, since it's my sort of introductory talk, I will just give you a very brief slide about what else my lab is working on, apart from what I will be talking about. So what I will be talking about are microbes and resources and how they compete for resources, how they evolve, and so on. But the other part deals with bacterial and phages. And we actually, right now, maybe we'll have a little bigger fraction of this phage-bacterial interactions occupying, a bigger fraction of my time will be occupied by looking at bacterial phages, because we just got awarded some RPH project, which means that we will have to do some milestones and deliverables dance, which I personally hate. I moved to biology about 25 years ago, and it was a time when the systems biology was just starting. So I was kind of doing this networks, gene expression, and so on. And after about 10 years, I got really bored with it, and I didn't do it for a while. And then when I came back, things changed. And what actually happened is that this systems biology became really predominantly a single cell studies. So these are fun. So I kind of started doing it again with the help of one of my students. So in particular, we developed some algorithm about how to infer RNA velocity, which is a change in the gene expression in a given cell, without using splicing variant. Yet, just to continue my spiel. So they are diverse, and yet on a certain level, they are universal. And again, this is also a little bit of a mystery. I will show you one kind of aspect of this universality and maybe one explanation explaining this universality in certain communities. What do I mean by universal? Well, you can have all kinds of zoo at the level of the species strains with weird names. But if you look at their metabolic functions in the right way, in the right angle, then you will see that it's the same. It's the same in different instances of this community. So something at least is universal. And without universality, really it becomes a stamp collecting. Now, the second kind of group of questions, stability and control. Microbial communities routinely have more than one stable state. We as quite a kind of engineers want to keep them in a desirable state, avoiding undesirable states. So how do we control these transitions between states? How we ensemble a community to make sure that it ends up in the right state with the right set of species in the right abundances? That's the second group of questions. And the last one is, of course, evolution. Evolution is really, really fascinating, but what is special about evolution in microbial communities is that it's a co-evolution, right? So you need to figure out how microbes in the presence of other members of the community evolve in a different way than they would evolve alone. I will kind of try to illustrate, again, I have tons of models in all of those aspects, and not only models, but also collaborations with experimental groups, some machine learning. But I will try to use just two vignettes, two models, to show you two aspects of those, of my work on microbial communities. So I will start with exhibit number 1, which will study how microbial communities can grow in a very energy poor, energy limited environments? And the answer is very slowly. This is a work which was driven primarily by my postdoc at the time, Ashish Bina George, who is a research scientist at the Broad Institute right now, and my PhD student, Ton Wang, who just started as an assistant professor at Purdue University, was also a co-author. It was published in ISME a few years ago. So let me try to hide this. And this one. Right. So let me first show you some data. It's not what we are trying to explain. It's not in a slow growing communities, but I just wanted to show you a picture of what does it mean for a community to be extremely variable in terms of their species composition and yet universal in terms of their functional roles, metabolic functional roles. This is a work done by Luca and collaborators, and they studied those bromeliad plants, which in a jungle, and inside those bromeliad plants, I can show you where exactly, somewhere here, there is a microbial community. There is a pool of water, and inside this pool of water is microbial community. And when they studied 33 of those bromeliad plants, and they kind of looked at what kind of species, genera are present, they saw a complete mess. They saw absolutely no rhyme or pattern in who is present, who is absent. And yet, when they kind of broke it down into metabolic functions such as fermentation, high-level metabolic functions, fermentations, oxygen respiration, carbon fixation, they saw some similarity between all those 33 pools. So this is, this is not alone. This is not just limited to bromelian communities. It is present in many, but not all, microbial ecosystems. And what we try to explain is what might be the mechanism behind this metabolic universality, metabolic convergence in slow-growing communities. All right, why would you even care about slow-growing microbial communities? Well, they are actually surprisingly abundant. We don't see them because they are deep, deep underground, under subsurface, under the ocean, in deep soils, and so on. But if you integrate the biomass, they, depending on who you ask, they occupy between 5 and 50% of the total Earth's biomass. Yes. Bromeliad, yes. Well, not at this level, but the question about anal is an excellent question. In fact, Casey Huang and collaborators at some point published a paper debunking some of the claims about universality, and I was cheering it because whenever you present some universality without anal, it's nothing. But in this particular case, to my knowledge, it hasn't been debunked. Maybe I'm wrong, but again, what you're right that what you need to do is you just need to mix together completely unrelated microbes without them being limited to this particular type of environment and see what kind of fraction will be by permutation of respiration and so on. So coming back to the deep soils, so growth in the deep soils is also a bit of a mystery, because if you look at the something called cell turnover, which is different from the exponential growth of cells, doubling time. So in energy-rich environments, we all know that in the lab we can make E. coli double every 20 minutes or so, or if you are dealing with some very slow marine microbes, maybe they will take days to double, but it's still minutes to days. Whereas if you look at a cell turnover, which means for how long a particular organic material stays inside a given organism, it can last for hundreds to thousands of years. It has been documented in multiple of those subsurface environments, and what presumably is happening is that most of those microbes are spending considerable amount of time in a dormant state, and every now and then they just wake up and repair a little bit of their machinery, because you cannot do completely dormant forever. And so But the bottom line is whatever happens, the growth or steady state maintenance in those communities is extremely slow. And in part it is explained by extremely low free energy of the reactions they use, but not only. Of course, the availability of the nutrients is another matter. And also, energy limited environments are also present in some human-made systems.
Is it like a witness or just a gradual change?
You mean like in the main use?
I mean, this energy rich versus energy limited and the time scales associated.
It's a gradient, definitely. You can literally look at it as a gradient. If you go deeper and deeper in soil, the less and less of organic matter sort of penetrates. And of course, if you are at the bottom of the ocean, you're already anoxic, so you don't have access to the oxygen. But the question is how much of you have this marine snow, which falls on the bottom of the ocean, which provides food for anything which lives in the water column. But if you're deeper, then you have to be more inventive in looking for your food sources. So again, quite frankly, not a lot of it, people don't know a lot about them because it's very hard to study. You need to drill and sample them from mostly the oil people, oil explorers drill, and then the microbial ecologists piggyback on this, oil people to share samples. But I'm saying that maybe this is kind of an extreme. I just wanted to give you this idea that sometimes it's really thousands of years. But in a more kind of human scale, that will not be a thousands of years. That will be still slow growing and energy limited, but wastewater treatment plants, landfills after the initial kind of easy pickings were done, and so on. So anyhow, let's move to the model. My models are kind of stylized and simple, but hopefully they can teach us something. So here is a model we developed with Ashish and Tong. We assume that there are certain substrates, which we call them R0 to Rn, and they are kind of arranged on a hierarchy of energies. This is most energy rich, second, third, and fourth. Now, they are all can be interconverted to each other. Right, so assumption is that any of the resource can be converted to any other resource, but of course, only conversions which are meaningful from the standpoint of microbes are the ones which convert higher energy to lower energy resources. Now, we also need to specify the species which consume those resources. Each species randomly selects a subset of resources which it can consume, and to each resource is assigned a number, which is called, we call it enzyme budget. Enzyme budget utilizing this resource. And the overall, actually the technical way how we do it, we first assign overall budget. Like imagine that it's a grant proposal. You have a budget and different microbes have different budgets. So it's not like all of them have the same budget. But then a microbe divides its budget between a certain reactions. Some of the reactions are not included at all. But I guess the central feature, which is really where all the fun things in our model are coming from, is this assumption that the reaction follows this reversible Michaelis-Menten kinetics with thermodynamic constraints. And this part, again, those of you who know a little bit of the systems biology or even chemistry know the Michaelis-Menten in this form. And this thing is added when reaction is reversible and when the products, when they accumulate, start inhibiting the flow through the reaction. And this form of this term is completely universal. It's dictated by physics. It's not dictated by biology or even by chemistry. So it is physics, thermodynamics 101. This P over S in particular, if you convert them into delta G, they become the entropy terms, which are, as we know, universal. So then what we do with this model? Well, we create first the environment with 6 resources, 15 reactions. Only the highest energy resources supplied externally. All others are generated as byproducts, but when they are generated as byproducts, they are being consumed by macro. Yeah.
Is there a sense of the reactions are near or the delta? In what sense is it actually slow?
Okay, very good point. So in our world, they are small because all the reactions are near the equilibrium, in the sense that the small number here comes from this 1 minus p over s exponential to g being very close with you. So reverse flow is always equal to forward flow. And well, you may kind of think about it.
Delta B relative. It's kind of statement about the delta P both.
No, no. But of course, the only way you can reasonably make it if your delta G zero is not terribly high, right? You cannot make it this aerobic reaction because then your P over S product over substrate needs to be, I don't know, 30 orders of magnitude, which is crazy. But for low energy delta G zero, you can have the situation where reverse flow is always equal to the forward flow. And you may say that, again, our microbes are not that far from being dead, right? So that being that the thermal equilibrium when the reverse flow is equal to forward flow. They are not completely dead, but the imbalance between forward and reverse is very, very small. Okay, so we generated a huge number of pools, well, 5 pools, but we seeded them with 600 species. All species were selected randomly with random enzyme budgets, with random parameters of the reaction, such as Kcat and Km and Ks. Of course, what was universal is this part. This part is coming from physics, so it is shared between the pools. Yeah, and in order to make sure that the growth is actually really slow, we made the communities to grow in a chemostat, which has this parameter called dilution rate. So in a steady state of a chemostat, all the surviving microbes will grow at exactly the rate of dilution. So if I make it dilute, turn around in a chemostat once every 10 days, then the average growth rate of microbes in my chemostat will be, once every 10 days. So that's how we make them slow growing. And as such, we kind of got a small parameter in our model, delta over G max, right? Delta is the real growth rate of microbes in our chemostat, whereas G max is a maximal growth rate which they can get if they are given plenty of resources and no product incubation. Okay, so here's what emerges. Here is what emerges from our model. Yes?
Your control S over P ratio.
No, I'm not a satellite cortisol. And that is exactly what happens. The reactions accumulate product until it slows the reactions down to the extent that that it matches the dilution rate of the. Okay, that's excellent question. So I'm not controlling anything except for the influx of R0, which doesn't matter. It's one of those parameters which I don't count. And well, delta over G max is my small parameter. And of course, thermodynamics is dictated by physics. Actually, not entirely by physics. There is a little bit of biology here. When you look at what happens in a given reaction, you may say delta G0 will be the free energy difference between the molecules, say R0 and R2. But in reality, some of this free energy is converted to ATP, and therefore it's not lost to dissipation. What delta G0 is characterized is whatever part of the free energy which hasn't been converted to the energy currency of the cell, which is a little bit depends on biology. That's why you don't have this. But again, this is quite universal. The same pathway will produce the same number of ATPs. Of course, if the microbes will use different pathways to do this conversion, they will be treated as different pathways. So here is what we saw. Here is just an example of 2 pools, pool 1 and pool 2. Out of 15 reactions, only 5 reactions survived. And the fluxes through those reactions, which are sizes of those circles, are the same. However, who is exactly doing the job in different communities, just like in this taxonomically diverse communities, is completely random, right? And the microbes are not specialists. For instance, this purple microbe is involved in 2 reactions. The green microbe is involved in 3 reactions, and so on. So it's somehow divided between the microbes while making the overall metabolic fluxes through the community conserved. Yes. Well, nobody measured. I don't expect this model to be applicable to bromeliad because they are definitely oxygen rich and so on. And I don't know. Well, I will show you in the very end. There are some experiments, not in the wild, but in industrial settings of the slow-growing communities, where people actually saw universality in resource concentrations and in, well, I don't think they measured fluxes, but the resource concentration with Miss Borer liver. So
How do you to connect Indians mama kid with the extreme?
oh yeah, that is definitely true. That is absolutely true. The microbes are very rarely specialists, so they are definitely having multiple pathways, yeah, yeah. One species is involved in multiple things, so you can kind of for each one of those flux, if you want, you can see how it is divided between different bacteria which contribute to this flux. This is overall community flux. Okay, so again, functional convergence, taxonomic divergence, I already talked a lot about. Network structure. Interesting story is that the topology of this flux network is that each product here is produced by only one reactions. So each one has in degree equal to 1. Out degree can be anything, but in degree is only equal to 1. So it turned out, after we kind of meditated on this problem, we figured out the math behind it. And the math was actually remarkably physicsy. So we found that the steady state ratios of resources in the environment follow the Boltzmann-Gibbs distribution up to a small parameter, right? So if delta over Gmax is very small, then the ratio of resources is basically delta G divided by RT. Okay? And it turned out that the whole thing organized itself, and we understand why, to a state where the dissipation rate through every reaction is at its maximum. So the reactions which are being selected because they are the ones which generate a given result with a maximal dissipation, the maximal delta G0. And again, delta G0, as I said, is not at all a fixed thing. It has a little bit of a biology in a sense that you can convert a lot of this energy into useful molecules like ATP, or you can convert less of it. And the more dissipative you are, the less you are prone to product inhibition. And as a result, the ecological competition ensures that if your higher dissipation pathway is present, it drives the resource concentration to the levels where the species using lower dissipation pathways cannot invade. So that's kind of the reason why this whole thing happens. Yes. It had been, I mean, this is a buzzword. If you look at the literature, there are plenty of papers which argue that there should be a maximal dissipation principle. And we did a literature review and nobody actually justified it, right? So we, for the first time, in a very, very limited setting, justified it and saw that it actually can be derived from first principles. And those are principles are ecological principles in our case, not. So I wouldn't expect it to happen, or at least I don't know how to derive it for single microbes sharing pathways. Okay, so this is a lot of blah, 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 except that we can calculate the fluxes and reactions. And that's why when we kind of look at the experimentally measured fluxes, experimentally means in silico, and what we predict by our theory, they agree very well. But second, everything is only up to the small parameter. The moment you start making the dilution rate, the growth rate of microbes to be comparable to the maximal growth rate, this universality starts to get broken. And we attempted to illustrate it here. And again, it's not easy to read. So just what we are showing is that the same community sampled from the same, assembled from the same pool sampled at slow growth rate and let's say at fast growth rate. And the reactions in one community are represented as solid arrows, in the other community as dashed arrows. What you should be looking at here is that the dashed arrows and solid arrows are precisely the same, but when you are at a fast growth rate, actually there is some idiosyncratic things start to intervene, right? Some microbes start using reactions which are not maximally dissipate, and just because they can get the better growth out of them through their parameters. And again, we kind of quantified how similar, up to maybe a few percent, when the microbial growth is up to a few percent of the max growth rate, the universality at the network level is 100%. But then it starts to kind of deteriorate, and by the time you are at, I don't know, 10%, it's already only 60% universal, and so on. So again, experiments are scarce. We managed to find a paper which studied sort of the anaerobic digesters in the lab, but seeded with different microbial communities, meaning that it's the same setup as in our paper, different pools, but occupy the same thermodynamic environment. And what we see here is the degree of when these numbers here are small, that means the community is universal. Here it is in terms of variation of metabolite concentrations, and the other one is metabolic distance. I forgot what it was, something like something else. But in all cases, there were kind of actually, I see. This is just for individual kind of metabolites. Acetate, propionate, butyrate were much more similar, where the dilution rate was slow, something like once every 10 days, maybe even slower, because this is delta per day. If it's 0.05, then it's a turnover of the chemostat once every 20 days. But when it was like once every 2 days, that it was kind of poor. So again, this is the only kind of piece of evidence we see that what we are saying here actually is operational in some communities. So there are a few take-home messages which again, maximum dissipation principle, if you are a big fan of it, and I knew some people who were kind of making it their main kind of scientific direction in their labs, but we have a way to derive it using physics, not ecology. Actually, using ecology and physics, not hand-waving. Now, the part which I still don't understand is how come that if the selection is for more dissipative microbes, dissipation is wasteful. If you dissipate everything, you do not grow. What prevents microbes from dissipating everything they have? And of course, at some point, making delta G larger beyond a certain scale stops being very important, because you already have a sufficiently large Gibbs factor when your delta G is large enough. So there is a diminishing return here. If you are dissipating progressively more and more, your competitive edge is getting less and less. And that's probably what is stopping what's stopping our microbes in our model from wasting everything. Okay, so I have another story to tell, and I hopefully have about 20 minutes, a little bit more, because I started later. So again, the same people are involved, except that this time Tong was in a driver's seat and Ashish was in a passenger seat in this project. It is actually a project, even though we started it a while ago, but it's right now finally got to the level where it's accepted in cell systems and should come out in the next months. So this story deals with something called auxotrophs. Auxotrophs are bugs which cannot on their own synthesize at least one essential metabolite. Think of amino acids, some nucleotides, some essential vitamins. We ourselves are auxotrophs. Without what we get from food or from our microbial colleagues, we would not be able to live. So we are not capable of synthesizing a couple of amino acids. I forgot which ones. And auxotrophs, surprisingly, are very, very prevalent. So if you sample microbial communities in the wild, you get 76% of them will be auxotrophs in at least one amino acid or one essential product. And of course, if you are sampling some microbes which live inside insects and the symbionts, they are basically gave up. They outsource to insects 21 essential products. But the free living ones, the ones which live in sort of in the wild, and the ones which live in our gut, are typically missing one or two. So, and the question is why? Because it, at the surface, it looks like not being able to synthesize something on their own violates this cowboy attitude that I have to do everything on my own, and yet they are everywhere. And what makes this tick? So we have at least an angle on answering this question. And once again, it is based on a model. So let me walk you through our simple model number 2. Again, the only similarity here is that the notation, the resources are still labeled R0 to Rn. Everything else is completely different. So forget about free energy. What is happening in this model is that one resource, which you can think about as glucose, for instance, R0, is being provided externally. And all of the bugs have a different capabilities. We call them transformation capabilities of converting this glucose into an essential, think about them, amino acids which are necessary for growth. So each microbe is characterized by a vector, n-dimensional vector of transformation capabilities. It's normalized to 1, meaning that kind of 1 mole of glucose will generate that much of this amino acid, this much of that. And of course, if you are an auxotroph, your corresponding projection on this vector is equal to 0, right? If you are an auxotroph on 2 amino acids, it will be 2 components of this vector which are equal to 0. Now, after making those amino acids internally, they all combine them in a biomass. And here is our assumption number 2, is that all of the microbes use the same stoichiometry in which they convert amino acids or nucleotides or whatever to their biomass, which is probably not too far from the truth. We also know that if we will start violating this condition, the whole hell breaks loose and communities become multi-stable, chaotic, and for those of you who like the twisted things, I'm be included. I have another model where I specifically made the stoichiometry different, and then we had another fun games. But here, all of the stoichiometries are the same, and by just changing concentrations, you can make it 1 to 1 to 1 to 1. Okay, so the way how microbes use the biomass precursors are the same across microbes, and they are given by 1 to 1 to 1 ratio. And then, of course, the real kind of growth is determined by the Lidix growth of the minimum, meaning that whatever flux you are getting for a given resource, which could be your own synthesis or something which you get from the environment, divided by stoichiometric coefficient, whichever one is limiting your growth, whichever one is in shortest supplies, that's what set up your growth. So if your limiting amino acid is number 1, you can give this bug whatever flux of amino acid number 2, 3, 4, and 20, it will not grow any faster. That's why the mean function here, okay? And this mean function actually is, is nice. It gives you enough nonlinearity for this model to be fun. Because a lot of those microbial communities models, they have some linear structure, and workable Terra including, or so-called MacArthur model. And again, they are fun, but they're not as much fun as when you have a nonlinearity using the mean function. Now, final assumption in the model is that whatever you don't use, you secrete. You excrete to the environment, not because you are so good and so nice to your neighbors, but because otherwise you will explode, right? So each bug is set at a certain limiting amino acid, which it limits its growth, and everything else which it produces in excess of this limitation is being excreted to the environment and consumed by others, okay? So altogether, those are my equations. In fact, maybe there is one more equation about what is being excreted and how. Yeah, that's if you are just a mathematician who likes to look at equations, you just look and then they have this on this set of 4 equations. But it turned out that the model actually has a very nice geometric solution, geometric interpretation, which I personally, yes.
Relative to the
Well, it depends on how well balanced are your TIs. I will show you, this is a model which we actually applied to the experiment where 14 auxotrophs were generated by their church and team follows. And why we go? We have mutants. They're not but they still had, even though they were synthetically made auxotrophic, they also this auxotrophy made them to vary their perforation capability. Because once you mess up one place inside, everything else is going to do. In the wild, probably those perforation capabilities are quite different. Again, I don't want to go too deep into it, otherwise I will be doing flux balance analysis and I hate flux balance. The one thing which I hate absolutely, and the irony is that this is a model which comes close to flux balance analysis, but at the community level, and it has a fun component, which is not. So I will show you first the flux balance sort of part. Let's see if I can do it like this. So here is one rule. So imagine that imagine I have a community with 3 resources, and each bug can be positioned inside this simplex according to its transformation vector. The way we choose to do it is like this. Those are orthogonal to the sides. So this bug, the red bug, has the T1, its transformation capability for R1, which is very small, meaning that this guy definitely will be limited by R1, T2, and T3. And then I can have another bug, maybe it will be a green bug, somewhere here, which will have a different sort of set of transformation capabilities. And maybe I will have another bug, a blue bug, which will be yet another vector of transformation capabilities. So the rule number 1, which is actually basically a flux balance analysis for communities, is that in order for this community of 3 bugs to survive, we need to ensure that the center of the triangle is inside the triangle formed by those 3 transformation capabilities. It's nothing else but just saying that community as a whole needs to balance its production. So you can, in fact, once I know the positions of those points, I will know the relative abundances of bugs in a community, and that is neat because it kind of gives me the predictive power. I'm erasing it because I want to draw another set of triangles, which would be hard to draw if I make this one messy. So the rule number 1, convex hulls of transformation capabilities have to have the stoichiometry vector inside. And if this was all there was, I would kill myself, because then it means that I just reinvented flux balance analysis. But, and I need to mess up a little bit, sorry. In my kind of attempt, I didn't place them well. I hope I will place them well now. Let's say, okay, this is still good enough for flux balance standpoint. But now comes the hard part. Now comes the ecology. I already told you that by Liebig growth of the minimum, each one has to be limited by 1 resource. In this full diversity community with 3 bugs, each one has to be limited by its own resource. If 2 bugs are limited by the same resource, one of them will necessarily be better than the other, and a better one will outcompete the hell out of the weaker one. So if you want them to live peacefully to each other, each one has to be limited by its own resource. For this, it turned out one needs to draw another triangle, which has oops. Let me try to draw it. It's first time I'm trying to draw it like this, like this, and like this. Okay, so what I try to draw are black lines are parallel to the sides of the big triangle. So it is also equilateral triangle. But each bug occupies its own side on this triangle. You see the green bug is on the side which is facing R2, which means the green bug is limited by R2, the red bug is limited by R1, and the blue bug is limited by R3. So if you succeed doing this, you are in business, because not only you can balance your resources, but you can also coexist without bugs outcompeting each other. So the ecology and the fluxes are both kind of satisfied. Okay, this is kind of my slides in case I would fail to draw them nicely. I think I half succeeded in drawing nicely. One thing which I didn't try to do is, so this triangle, the second triangle, which is drawn in light blue here, is actually has a property that any bug with transformation vector inside this blue triangle would be able to invade this community. So we did this little experiment. So we first made a community with bugs alpha, beta, gamma, and then we tried this cloud of dots, which are all kind of our other in silico bugs. And the ones which are marked in light blue invaded, and the ones which were outside were outcompeted by others, okay? Which is kind of, understandable. If you are outside of the triangle, means you cannot grow on a concentration of the resource which is currently controlled by other bugs. All right, so now why auxotrophs? I have actually I'm doing okay in time, which is surprising. Normally I'm running out of time. Okay, so I kind of posed one question: why auxotrophs are so widespread in nature? And we have, through the lens of this model, we have at least one tentative answer to this question. So in our base model, we didn't supply anything but the resource R0, glucose. In real life, the amino acids are coming externally. Maybe not everything which you have in your little pool is generated by your buds. Maybe some nearby buds also give you some of their resources. It turned out that in this model, this external supply of amino acids is very easy to account for. All you need to do is to shift your stoichiometry from 1, 1, 1 to another point, which is shifted slightly by external supply. It's a subtle mathematical point, but trust me, it is true. So what I'm showing here is a situation in which this community, which initially can coexist, this is community of non-auxotroph. They're actually called prototrophs, if I remember correctly. They can synthesize everything on their own, and they are kind of having this stoichiometry vector in the middle of this triangle. But if the external supply starts to vary a little bit, they are vulnerable to extinction, because the external supply will make this cross to drift off to a place like this, which will be already outside of the triangle. This is kind of confirmed by in silico simulations when we started messing around with external supplies, and after a while, just orange bug happened to be the only survivor. Now, this is a community of 3 auxotrophs, which are all missing one resource. And they have a much kind of more stable triangle, which is much more stable to external resource fluctuations. And here we have 2 survivors. But if you will make, in this kind of toy model, a community of double auxotrophs, actually each bug here will be a specialist only capable of producing one of the 3 resources. All 3 of them will need each other, and they will survive at whatever external supply rate you can. Again, it's a nice way of quantifying what is ecologists are called structural stability. Structural stability is how stable your community is with respect to resource fluctuations. Here is just the geometric factor, the ratio of the yellow triangle to the big white triangle. That is more or less your structural stability, measure of your structural stability. Okay, so that is kind of point 1. And interestingly enough, there is an indirect experimental observation, which again is pretty lousy. If you ask me, the correlations are 0.27, 0.14, 0.13, but statistically significant and reproducible, which is very important, because a lot of times in biology, you see it in one study and you don't see it in another study. At least this one, you saw it in 2 independent. This is gut microbiomes. What is plotted on the x-axis is actually a difference here. So here, y-axis is how many auxotrophs weighted by their abundance are in community. And x-axis is the diversity of a community, and y-axis here is the stability. So the bottom line is more auxotrophs you have in the community, the more stable the community tests. Okay, take it at for whatever it's worth. At least this is the communities in the wild. But now, what my kind of piece de resistance, which I would say is actually explained in an experiment. It's an old experiment. You see, this paper is more than 10 years old. And what those guys did is they took good old E. coli and they knocked out some pathways, making it auxotrophic. And they managed to make 14 auxotrophs out of 20 possible auxotrophs because they're 20 amino acids. Now, somebody in this community was very fond of Lotka-Volterra models. And again, nothing wrong with Lotka-Volterra models, but sometimes you want to go outside of the Lotka-Volterra model. So in the next few slides, I will be trashing Lotka-Volterra model. If you're a big fan of Lotka-Volterra, step out and walk back in, okay? So Lotka-Volterra, they made a pairwise fits with Lotka-Volterra model. And of course, you have as many parameters as you have data points. So it's not surprising the fits were good. And then they did a separate experiment, where they put all 14 members in one pool. It was actually not a chemostat; it's a serial dilution experiment, but it's the same thing. So, so in the end, after diluting it for 50 days, 50 daily dilutions, they had only four auxotrophs out of 14 that survived. Okay. So now let's compare. So we have two models with the same number of parameters. So it's apples to apples comparison. In our model, we were fitting based on the data, those transformation vectors, T1, T2, T3, and so on for each of the 14 strains. So we had 14 times 20 parameters, something like this. So here's our model. AM is our auxotrophic model. Here is generalized Lotka-Volterra. And generalized Lotka-Volterra fits pairwise experiments. And what is a pairwise experiment? They put just two bugs together. They let them grow until they coexist, and all of them coexisted. And they predict the relative abundances of coexisting pair. And they did it perfectly. I mean, they got an absolutely A, and we get, if I'm really generous, we get B minus here. Okay? So far, we are on the losing side. But now comes the pièce de résistance, the 14 bug experiment. As I already told you, experiment done in duplicates. So there were two biological replicates of the experiment. 4 bugs survived in slightly different relative abundances. Now, Lotka-Volterra tells all 14 will survive, which is F. This is no doubt. I mean, this is as bad as you can predict. We predicted 3 out of 4, which if I'm being generous to ourselves, I will call it A minus. If less generous, I will call it a B. Okay, so we kind of predicted 3 out of 4 survivors in this community of auxotrophs, which is definitely better than predicting 14. We also understand more or less what is going on. At least, especially easy to explain why delta M and delta T are so good. So this is our fitted matrix. So those are amino acids, 14, and those are strains. Delta is a standard biological notation for something where a particular amino acid has been knocked out. The pathway producing amino acid has been knocked out. When you see blue, that means that according to our fit, this strain produces a particular amino acid above average. When you see red, below average. So what you really need to have are, and of course, delta M, of course, on the diagonal, you have deep red because the auxotrophs don't produce at all the amino acid for which they were knocked out. Now, what you need to have is a pair which is producing above average the amino acid which the other one is missing. And the best pair like this, which we can just independently study this table and find that the best pair like this is the delta M and delta T mutants. Delta M actually produces a lot of stuff, including T, which delta T needs. Delta T only overproduces M and S, but M is what delta M needs. And again, for more subtle reasons, delta K also enters into this trio. We don't understand what is behind this one amino acid, which we cannot predict. Okay? So the bottom line is auxotrophy may be not a bug, it's a feature. It may be an evolutionary strategy. And higher order interactions is another buzzword in ecology. Our model definitely is reaching higher order interactions because of this nonlinear mean function. And Lotka-Volterra is, by definition, has no higher order interactions. Everything is pairwise. And pairwise is really useless when you are trying to predict these essential resources. Don't take me wrong, Lotka-Volterra and its analogs are probably still pretty good in predicting growth of substitutable resources. If you have the different carbon sources, Lotka-Volterra would be quite reasonable way of approaching it. But when you are dealing with multiple essential resources, the message of this talk is that it will suck. Okay. And just on a joking side, I say that I did show you two models which kind of follow my philosophy. And my philosophy is that I don't like top-down and bottom-up model because I feel like this is just the BS which you put in the grant proposal, top-down, bottom-up. What I like is a bottom-down model. What is a bottom-down model? You take a model which is already simple enough, and then you kind of strip it off parameters, hopefully keeping the interesting parts in, and then you are being graded. You see, I teach, so I'm obsessed with grading. So if you are a zero parameter model, then you get A+ and Per Bak Memorial Award. Per Bak was my PhD advisor, who was also very, extraordinary bottom-down modeler. And then the grade decreases with number of parameters, 5 or more, you get F, but don't despair, you can always try neural networks, which I also do in my lab. All right, take care.
Very tight. But I
Well, actually, 74% of the or something like this is auxotroph. But keep in mind that it's on the I managed to brainwash you to say that it's nice, but if you're living alone, and you're an auxotroph, you're in deep trouble. So you should kind of weigh in how you as an auxotroph can make a particular turbidity stable against resource fluctuations. This is what I really write in this starter here. And what you will do if you will find yourself alone, right? So it's kind of a balancing act.
And like the main argument to that is this ability that me that it times and get to the race model demographic models in the model. What happens if there's an exclusion of one of the species and what's with us?
Well, actually, in the model, when we simulated it, there is a demographic. In simulations, there is a demographic noise, and that's why this. Well, here there was. Here, I believe what Tong did, who did the simulations, is combination of those resource changes in resources as well as demographic noise. Of course, demographic noise only matters when you are really, really low in abundance, and then it can drive something to extinction. In our model, unless we kind of specifically happen to select something which is where one species is almost negligible in abundance, it will not be the case. By the way, there is a nice geometry here, which I haven't thought much about it, I must confess. But if you have a triangle, and if you're one of the species, if this one of the lines is very, very close to the stoichiometries, then one of the resources will almost not be needed, or one of the species will almost not be needed, and this one will be actually in a low abundance, and that is the one which would be vulnerable to stochastic fluctuation. This is actually a nice little follow-up which one could do.
You would have had the dissipation argument for that. So, am I understanding it correctly to say that the low energy the system is going to a state where the product inhibition is at its minimum? That product inhibition is low, but the space is on the ether.
I hope this one will explain it in a language which we both understand. So, for instance, this ratio of steady state resource consideration is tied to the exponent of delta G0 over RT. What we are comparing, if the same, imagine that one of those resources, say Ri, is the externally provided one. But the other one would be the more dissipative is the pathway. The dissipative pathways have a more larger negative delta G zeros, right? They are more irreversible, and they will make the downstream resource to be present at a lower R star. And we all know from ecology, the lower if your R star, in standard resource driven ecological models, the lower is your R star, the harder it is being laid. So
And this doesn't, and then the departure high energy.
Well, yeah, it is elegant, but in reality, again, this is so the bottom line is okay. Maybe the right way of thinking about it is here I'm ignoring the variability in those three factors of the mechanism. I only focus on this 1 minus P over S. But in reality, a bug which has some, by pure chance, has a large K cap and other parameters can partially compensate for a little bit of this, and this will only matter at high. I cannot really tell you better intuition than this. Just that when you start sort of writing down the equations, those equations do not emerge for free like they would be in a Gibbs free energy. Because in the end of the day, you still need to say that every bug delta has to be equal to its growth rate, which is the sum of the flux through its reactions, 1 minus exponent P over S exponent delta G over RT, sum over different reactions. So that is what you need to solve for. And this being small means that the usual kind of, if you can entirely, if I haven't thought about it more about if I make every prefactor in every reaction the same, that it would be always true. This is my kind of guess right now. But the fact that there is this idiosyncratic variability between microbes is what making it upon the atrocity. Very question.
So like sometimes you can model into the final interaction in local grammar by adding the quadratic terms, and non-linearities can be casted to this type of terms. So I was wondering if you could think of what would be the order of the terms involved this non-linearity.
Yeah, I haven't thought about it, but we will talk about it right after, because I guess we are meeting at 11, right, or 11 something. But I haven't thought about it. But of course, putting them into local term means that you're increasing the number of parameters, right? And correctly so, because the quadratic terms are the square, cubic terms are the cube, and so on. So I hope that, I haven't thought about how to, because there are two things which I try to stay far away from: local term and flux balance analysis.
I have a follow-up question on Verika's question. So like when you're like showing the comparison between Lotula and your model, are you using like the model with stable gravity?
I don't remember, quite frankly. But again, I don't believe that stochasticity, unless you are in one of those very exotic situations where one of the lines come close to stoichiometry, plays a major role here. So I may be proven wrong. I know that Nigel and you have worked a lot on stochasticity. So everybody is kind of looking at things through the things they work on, and I bear my own biases. So I believe that again, Tong is a competent simulator, so I believe he used the stochastic equations because he wanted to simulate the disappearance. Well, you can disappear if you are outside, you're just exponentially dying off. But at least he knew that, okay, stochasticity might be at least present in the following sense, that when a species abundance exponentially decays, once you cross the abundance of half a single microbe per chemostat, it's gone forever. You can do a little bit better by saying that at this point it can be maybe disappear faster.
Any more questions? So if
Anybody from Zoom? Maybe I see Q&A here. Okay. Can you fix the idea works? Okay, that was old stuff. Well, that's rightfully so, because it's really boring to listen to talk that's happening on the
like so let's thank Sergei again.